Hello, everyone, and welcome to today's webinar. I'm Christina Jewell of LabRoots, and I will be your moderator for today's event. Today's presentation, how can clinicians use spatial transcriptomics data to interpret the complexity of tumor microenvironment, will be presented by Dr. Kwan-Jun Na, Chief Medical Officer, Portray. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more, please visit vizgen.com. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. Dr. Na, welcome. You may now begin your presentation.
Well, thank you for the kind introduction. Hello, I'm Kwan-Jun Na, the Chief Medical Officer at Portray and an Associate Professor in the Department of Thoracic and Cardiovascular Surgery at Seoul National University Hospital. I am honored to be invited to this meaningful event, the LabRoot webinar. I mainly perform surgeries for thoracic tumors like lung cancer and esophageal cancer. In my request to understand the cancer better, I have focused extensively on the tumor microenvironment. This interest led me to explore spatial transcriptomics, and today I will introduce the methodology and research application using platforms such as MERSCO. The topic of my presentation today is how clinicians can use spatial transcriptomics data to interpret the complexity of the tumor microenvironment. This is my disclosure. I'm a co-founder of Portray. First, I'll briefly explain why spatial transcriptomics is used in cancer research and introduce various methods that have been developed as spatial transcriptomics rapidly advances. In particular, I'll focus on image-based spatial transcriptomics. Second, I will discuss a recent study from another research institute that utilized MERSCO for lung cancer research. This study was aimed at finding crucial clues for immunotherapy. Finally, I'll explain the methodology developed at Portray for applying and analyzing spatial transcriptomics across the diverse range of patients using tissue microarray. I will also present one of our studies that employed MERSCO technology. Well, first, let's explore the usefulness of spatial transcriptomics in cancer research and delve into image-based spatial transcriptomics. As we all know, the ways in which we can obtain information about genes have been constantly evolving. It was only in the mid-2010 that it became possible to obtain transcriptome data at single-cell level from bulk tissue transcriptomics. In recent years, technologies that can provide spatial context in addition to the single-cell level data have been released. And now we have established new technology to interpret single-cell level data along with spatial information. Then why should clinicians focus on spatial biology to understand cancer? I believe it all comes down to the tumor microenvironment. Since the tumor microenvironment plays a crucial role in the development and evolution of cancer, understanding the composition and role of specific immune and stromal cells can aid in developing personalized medicine. Spatial information can provide many useful insights that cannot be inferred from traditional bulk sequencing data or single-cell RNA sequencing data. First, spatial biology allows us to determine the location information of microenvironment cells. For example, intratumoral fibroblasts and extratumoral fibroblasts might have different functions and exhibit different effects on cancer evolution. Second, we can perform cell-cell interaction analysis using spatial proximity. As cells interact with neighboring cells, this information can be useful in understanding how cancer cell interacts vary according to clinical characteristics. Third, spatial biology enables the analysis of ligand-receptor interactions. So this information may aid clinicians to predict treatment responses and prognostic stratification. Lastly, we can identify distinct spatial features within the tumor microenvironment, such as tertiary lymphoid structures and some immunity hubs. As you can see in this slide, spatial transcriptomics enables diverse analytical approaches to deeply understand the cancer microenvironment. Using RNA sequencing data, we can perform clustering and network analysis to simply characterize each cancer microenvironment. Based on histologic image data, we can also deeply analyze specific cell types and compositions within regions of interest. Additionally, developing functional scores from RNA sequencing data is also possible. By using co-localization and communication analysis, clinicians can understand the complex cell-cell or ligand-receptor interactions for each patient. As you might know, there are two distinct types of spatial transcriptomics: NGS-based spatial transcriptomics and image-based spatial transcriptomics. NGS-based spatial transcriptomics, such as Visium, SlideSeq, and SeqScope, rely on next-generation sequencing to map gene expression data to spatial locations within tissue sections. These methods provide high-throughput data, allowing for comprehensive analysis of the transcriptome across large tissue areas. On the other hand, image-based spatial transcriptomics, including platforms like MERSCOP, Xenium, and NanoString Cosmex, utilize imaging techniques to visualize and quantify gene expression directly within tissue samples. These methods offer high spatial resolution, enabling the precise localization of transcripts at the single-cell level. Both approaches have their unique advantages. NGS-based methods excel in generating large-scale data, while image-based method provides detailed spatial context. In this slide, we compare two image-based spatial transcriptomics methods, Xenium and MERSCOP. These two methods have different mechanisms to obtain the spatial transcriptome data. Xenium offers high sensitivity and specificity with two-dimensional image capabilities. However, it can struggle with non-specific signals, particularly in brain formalin-fixed tissues. On the other hand, MERSCOP also provides high sensitivity and specificity, but it's the advantage of 3D imaging. The trade-offs include longer workflows and sensitivity to RNA integrity, which requires careful handling of the samples. Both methods are powerful tools for spatial transcriptomics, each with its unique strength and considerations. In this section, I would like to briefly show a recent study that used MERSCOP to decipher tumor microenvironment of lung cancer, especially the spatial features related to immunotherapy response. As immunotherapy have revolutionized the lung cancer treatment, and its mode of action is deeply related to the immune cancer cell interactions, many researchers use spatial transcriptomic technology for understanding the immunotherapy response and resistance. So this study, published in Nature Immunology earlier this year by Jonathan Chen et al., discovered novel spatially organized stem immunity hubs associated with the response to immunotherapy in lung cancer patients. The researchers acquired spatial transcriptomics data from multiple lung cancer patients who underwent immunotherapy, including 48 non-responders and 20 responders. Using pre-treatment spatial transcriptomics data, they investigated spatial characteristics related to immunotherapy response. In their previous research on colon cancer, they identified immunity hubs related to clinical outcomes. They applied similar concepts to lung cancer and identified a sub-immunity hub that was abundant in responder samples. So they also revealed that these patients with this sub-immunity hub had a good response to immunotherapy with better overall and progression-free survival. Therefore, they conducted a deep analysis of this subcluster using MERSCOP technology. Using MERS-CoV data, they performed clustering analysis using cell information and spatial tile information, identifying a stem immunity hub specifically in their data. This hub is distinct from mature tertiary lymphoid structures and is enriched for stem-like TCF7 positive, PD1 positive, CD8 positive T cells, activated CCR7 positive, LAMP3 positive dendritic cells, and CCL19 positive fibroblasts, as well as chemokines that organize these cells. These characteristics were distinct in responder patients. They also performed co-localization analysis and ligand receptor and interaction analysis using MERS-CoV data. They discovered that CXCL10 positive macrophages and TCF7 negative CD8 T cells were mostly co-localized and had strong interactions within stem immunity hubs. Moreover, a similar relationship was observed between mature regulatory dendritic cells and TCF7 positive CD4 positive regulatory T cells. These results provide a comprehensive picture of the spatial organization of the human intratumoral immune response and its relevance to patients' immunotherapy outcomes. So this study is one of the good example of how the spatial information and analysis from MERS-CoV technology can be used in clinical settings and can be identify clues for immunotherapy responses and resistance. In this section, I will demonstrate how we generate MERS-CoV data from multiple samples using tissue microarray and how we analyze them. I would like to present our recent data investigating the role of fibroblast in cancer progression of lung squamous cell carcinoma. Now, let's discuss some of the disadvantages of spatial transcriptomics. First, the cost is significantly high. This makes it difficult to analyze multiple samples, limiting the scalability of the research. Second, the data scale is often very large. Handling and analyzing such vast amounts of data requires substantial computational power, which can be a challenge with limited resources. A third, complex bioinformatics skills are required to analyze data. Applying the appropriate bioinformatics pipeline can be difficult, necessitating specialized expertise and often creating a barrier for many research teams. So we think that using tissue microarray, we can generate multiple cores from multiple patients in a single experiment. This approach allows us to collect comprehensive cancer data efficiently, addressing the high cost associated with analyzing multiple samples. By integrating tissue microarray with spatial transcriptomics, we can significantly reduce the expenses and streamline the analysis process, making it more feasible to conduct large-scale studies. So in Portray, we have also developed a downstream analysis pipeline for spatial transcriptomics data, which is acquired from tissue microarray. So in this slide, we are showing the example how we acquire the MERS-CoV data from tissue microarray and how we conduct the downstream analysis from this novel data. As you can see in this slide, our automatic method allows us to dearray the tissue microarray core by core, facilitating detailed analysis and reducing manual effort. On the left side is the raw data from MERS-CoV, and on the right side, our automatic algorithm divides the cores into separate clusters. So this allows us to treat the data from individual cores as the data from single patients. This automation further enhances our ability to conduct large-scale studies effectively. By using our algorithm, the individual small cores from tissue microarray were perfectly separated into distinct datasets, allowing each core to be analyzed as a unique dataset from a single patient. This separation enables us to characterize the tumor microenvironment of individual patients more accurately. Utilizing single-cell RNA sequencing data, we can annotate and analyze the specific immune and stromal cells within the cancer microenvironment. This detailed cell subtyping provides deeper insights into cellular composition and interactions within the tumor microenvironment, enhancing our understanding of cancer biology and potential therapeutic targets. In addition to detailed cell annotation, we can use this data for major cell subtyping within the tumor microenvironment. As shown in this slide, our analysis allows us to classify the major cell types present in a single core. So we divided the cells into epithelial cells, fibroblasts, myeloid cells, T and K cells, and endothelial cells. We identify and categorize cells into groups such as endothelial cells, epithelial cells, and others, so forth. And this classification is visualized in both UMAP plus and bar charts, so we can analyze which cells are abundant in this core and which cells are located near other cell types. So for this downstream pipeline analysis, we can simply understand how the tumor microenvironment is composed with a certain type of cells. So using this advanced analysis method, we obtain multiple merge-scope datasets from lung squamous cell carcinoma to conduct our own research. As many of you know, lung squamous cell carcinoma is an aggressive subtype of non-small cell lung cancer, often diagnosed at a later stage. So tumor microenvironment, particularly cancer-associated fibroblasts and immune cells, plays a crucial role in cancer progression and evolution. So we would like to analyze the role of the immune or fibroblasts within the lung squamous cell carcinoma and how they work for the cancer evolution and cancer aggressiveness. So in this study, we aim to address a critical problem. The number one, the spatial interactions, and number two, the roles of cells in tumor growth and patient prognosis in lung squamous cell carcinoma. And our objective are, first, we seek to dissect the spatial relationships of cell types and molecular features within the tumor microenvironment of lung squamous cell carcinoma. Second, we aim to identify associations between the spatial patterns and the progression of lung squamous cell carcinoma. Through this research, we hope to gain deeper insight into the complex interactions within the tumor microenvironment and their implications for cancer progression and patient outcomes. So this slide outlines the overall scheme of our study. So we started with reference cells obtained from single-cell RNA sequence data to create a comprehensive cell atlas. And using the publicly available algorithm, which is called TACO method for cell labeling, we applied this reference to our tissue microarray samples from 33 lung squamous cell carcinoma patients. So these are the all merge-scope data. We then analyzed the cell composition and spatial patterns of cellular distances within the tumor microenvironment. These spatial data were linked to clinical pathologic parameters such as tumor size, the FDG PET uptake, imaging results, and patient's prognosis. In addition, we examined the spatial gene expression to further understand the molecular characteristics within the tumor microenvironment. This integrative approach allows us to correlate specific spatial patterns with clinical outcomes, providing deeper insights into the role of tumor microenvironment in lung squamous cell carcinoma progression. So our findings revealed that tumor growth in lung squamous cell carcinoma is associated with cancer-associated fibroblasts and their spatial patterns within the tumor microenvironment. On the left side, You can see the composition of cells within the tumor microenvironment, highlighting the various cell types present within the single cores. The graphs on the right illustrate the relationship between the tumor growth and fibroblast proportion. We observed a negative correlation between fibroblast proportion and tumor size, as well as SUV mass, which is an indicator for tumor glucose uptake. The lower fibroblast populations are associated with large tumor size and higher SUV max, which is correlated to the higher glucose uptake. The heat map in the bottom left corner shows neighborhood enrichment depicting the spatial proximity of different cell types within the tumor microenvironment. The bottom right graphs further demonstrate the increased spatial closeness of fibroblasts to tumor epithelial cells correlates positively with tumor size and the SUV max. In summary, our data set suggests that while the overall fibroblast population decreases, the spatial proximity of fibroblasts to tumor epithelial cells increases, which is associated with tumor growth and progression. Our research indicates that the proximity of fibroblasts to tumor epithelial cells is associated with poor outcomes in lung squamous cell carcinoma. On the left, we have visual representations of fibroblast infiltrated, and fibroblast excluded tumors. The PET-CT images in the middle illustrate the difference in the tumor size and metabolic activity between these two conditions. Tumors with higher fibroblast infiltration on the top tend to be larger and show higher SUV max values, indicating increased metabolic activity compared to the fibroblast excluded tumors on the bottom. The Kaplan-Meier survival curve on the right clearly shows that the patients with fibroblast infiltrated tumors have significantly worse recurrence-free survival compared to those with fibroblast excluded tumors. So our data suggests that increased fibroblast infiltration into the tumor is associated with higher glycolysis, increased tumor proliferation, and reduced recurrence-free survival. These findings highlight the critical role of fibroblasts in the tumor microenvironment and their impact on tumor aggressiveness and patients' long-term prognosis. We conducted another study using NGS-based spatial transcriptomics Visium for how the fibroblast and the cancer epithelial cells co-localizes within the tumor microenvironment. Our analysis shows that fibroblasts and epithelial cell co-localized regions exhibit upregulation of glycolysis in lung squamous cell carcinoma. On the left, we have a spatial map, the Visium data, indicating regions where fibroblasts in green color and epithelial cells in blue color overlap in yellow color. This co-localization analysis suggests an interaction between these cell types that could influence the tumor metabolism. The dot plot on the top right displays the gene expression differences between the fibroblast co-localization with epithelial cells and those apart from epithelial cells. Of note, genes involved in glycolysis, such as NDRG1, SLC2A1, are highly expressed in the co-localization regions. The bar graph at the bottom shows enriched gene ontology biological processes with glycolytic processes prominently upregulated in these co-localization regions. This indicates that glycolysis is significantly metabolic pathway activated in the fibroblast epithelial interactions. These findings highlight the metabolic crosstalk between fibroblasts and the tumor epithelial cells. Our research also demonstrates the fibroblasts exhibit different states based on their spatial proximity to tumor epithelial cells in lung squamous cell carcinoma. The heat map on the left illustrates the distance of fibroblasts from epithelial cells, with warmer colors indicating closer proximity. We identified two distinct states of fibroblasts, epithelial adjacent and epithelial distant. The dot plot on the right shows the expression of genes associated with these fibroblast states. Notably, MMP111 is highly expressed in epithelial adjacent fibroblasts, while PDGFRA is predominantly expressed in epithelial distant fibroblasts. The images at the bottom further illustrate the spatial location of these fibroblast subtypes. The epithelial adjacent fibroblasts marked by high MMP1 expression are located at the periphery, whereas epithelial distant fibroblasts marked by high PDGFRA expression are found in the center of the tumor. And these findings were also recapitulated in the immunohistochemistry data, but I could not prepare the image for today's webinar. These findings suggest that fibroblasts in close proximity to cancer epithelial cells are in distinct state, which could influence their function and impact on tumor progression. Understanding these spatial relationships is crucial for developing target therapies that address the specific roles of fibroblast subtypes within the tumor microenvironment. So to conclude our findings, our research underscores the critical role of spatial organization and the molecular features of cancer-associated fibroblasts in influencing the progression of lung squamous cell carcinoma. The insights gained from this study highlight the potential of spatial transcriptomics in unraveling the complex spatial dynamics of the tumor microenvironment. This approach offers new avenues for targeted therapies that consider the spatial context of cellular interaction within tumors, ultimately aiming to improve patients' outcomes. So to summarize my lecture, image-based spatial transcriptomics, including MERSCOP, can decipher heterogeneous tissue microenvironments at cellular level, aiding clinicians in deeply understanding the characteristics of patients' tumor microenvironment. Prior research has utilized MERSCOP to comprehend spatial relationships and interaction between various cell types within complex microenvironments. Additionally, some spatial patterns have been revealed to have close relationships with tumor evolution and treatment responses. This understanding may assist clinicians in applying personalized medicine based on the characteristics of the tumor microenvironment. So thank you so much for your attention today. If you have any questions, do not hesitate to contact me through this email. Thank you.
Thank you, Dr. Na, for that excellent presentation. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted your questions, please do so now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So, Dr. Na, we already have so many great questions coming in, so I'm just going to dive in. Let's start with the first question here. How much has spatial biology impacted or advanced cancer research from a clinical perspective?
Well, that is a very great question. As the spatial transcriptomics research begins from 2021, as far as I remember, and many studies using spatial transcriptomics data are popped out within 4 years. So I can see so much the great research using spatial transcriptomics data. Well, the advantage of using spatial transcriptomic data using only single-cell RNA sequencing data is that we can use the H&E histopathologic image, which we can analyze the image patterns of the tumor microenvironment, and also we can get the spatial context of the tumor microenvironment, so we can analyze the cell network analysis and the cell-cell interactions. So well, using spatial transcriptomics data, we can analyze the the therapeutic targets for specific cancer or targeting some cells within tumor microenvironment. And we can also predict the therapeutics such as an immuno-oncologic agent which targets a tumor microenvironment. And also we can analyze some very specific molecules or very specific cell types that can modulate the tumor microenvironment during the cancer progression. So I think the spatial transcriptomic data research will be more released within the 10 years, and it will be the major topic for the deciphering the tumor microenvironment.
Excellent. Thank you for that answer. Our next question. Do you see more rapid adoption of spatial transcriptomics in clinical settings that other technologies like single-cell RNA-seq, for example?
Well, it is very hard to say because both single-cell RNA sequencing data and the spatial transcriptomics data is so expensive experiments in clinical settings. So patients have to pay a lot of money for this experiment. But I think spatial transcriptomics data have several advantages over single-cell RNA sequencing data in few aspects. So first, we could use FFP block data, which is archived in the pathologist department. But the single-cell RNA sequencing data, they need very fresh tissues. So we can use archived the tissue blocks for generation of a spatial transcriptomics data. So the application of this technology is more expensive than the single-cell RNA sequence data. And second, as I showed in the presentation, we could use a tissue microarray for a single experiment of spatial transcriptomics and can generate 15 to 18 patients' data at a single experiment. So single-cell RNA sequencing data and spatial transcriptomics has a similar cost for a single experiment, but single-cell RNA sequencing is for only single patient. But if we use tissue microarray, we can generate 18 patients' data from single experiment. So it can be cheaper per patient. And third, the time to generate the data is much shorter than the single cell experiment. So we could get data spatial RNA transcript data within a week for a single experiment. But for single cell RNA sequencing data, in Korea it takes usually a few weeks for we can get the data. So it is more rapid. And lastly, as spatial data can give us single cell level data as well as the spatial context data, so that if the automated algorithms apply to this data, we could get more complex results of analyzing tissue the tumor microenvironment than the single cell RNA sequence data only.
Very good. Thank you. Our next question. Are there therapeutic candidates that you will look at based on these findings?
Oh, well, so in this study, we focused on the function of cancer-associated fibroblast for the cancer progression and evolution. If there is some specific cancer-associated fibroblast that is associated with cancer growth and evolution, I think identifying the specific surface target on this fibroblast might be a good therapeutic candidate for prognostic prediction, as well as a candidate for such as radioligand therapy. As we did not investigate it further with this data for finding new targets, so I cannot show you the drug discovery process with this data, but I'm pretty sure that these findings might be a good clue for drug discovery process after if we decipher the specific cancer-associated fibroblasts for therapeutic targets.
Thank you. Our next question. Could I please ask how Xenium struggles with non-specific signals and how MERSCOPE can overcome this issue?
Well, this question should be asked to the vendors, and they might have more specific answers for this question. But as far as I know, Xenium struggles with non-specific signals due to its lower sensitivity in distinguishing between similar targets, so leading to background noise. But MERSCOP overcomes this by utilizing high-fidelity probes and advanced signal amplification techniques. So it results in the clearer and more accurate detection, as far as I know.
Very good. Thank you. Our next question. How do you distinguish cell types in the images? Is it possible to get the images with annotations?
Well, that is very great question. So in our company and for my research, I always try to get the spatial transcriptomics data and the H&E stained histopathologic image within the same slide. But for MERSCOP, we are trying to get the same histopathologic image from the MERSCOP slide, but we have not developed the protocol yet. But for Xenium, there's a protocol that we can get the histopathologic image from the same Xenium slide. So we can integrate the histopathologic image and the Xenium data and analyze it with together. So for annotating histopathologic image, we need pathologists to annotate the data. But if we integrate the spatial transcriptomics data and histopathologic image, then we can annotate the spatial transcriptomics data, and we can integrate that with the cell image of the histopathologic image. So integration can be done in the single cell level with the spatial transcriptomic data, and we can transfer it to the histopathologic image and use it for analyzing the image data.
Thank you. Now, Dr. Nam, do you think CAF's distribution could be affected by underlying lung diseases, such as COPD or interstitial lung disease?
Yeah. That's a great question. So COPD and interstitial lung disease, they are kind of inflammatory disease that will be progressed to the fibrotic phase, and the lung will be very fibrotic at the end stage of these diseases. So the CAF distribution might be affected by the underlying lung diseases. But as far as I know, there are many subtypes in the cancer-associated fibroblast, and the each subtypes of cancer-associated fibroblast has different functions for that contributes to the evolution and the cancer growth. So I think they might affect by the underlying lung disease, but if it comes to the cancer evolution and cancer growth, then some specific subtypes of cancer-associated fibroblast has their genuine function for cancer evolution.
And Dr. Nam, piggybacking on a question 2 times ago, are the annotated images available for the researchers?
Oh, so if we generate the MERSCOP data in our company, then we can give you the integration of image and the annotated spatial transcriptomic data together. So we cannot give you the annotation of by pathologist, but we can give you the single cell level annotated data of spatial transcriptomics and the corresponding image together.
Now, Dr. Na, what stage of LUSC samples were used in your study?
Oh, so that is a very great question. So I'm a thoracic surgeon, and I do the operation for the curative intent for the lung cancer. So most of the lung cancers are stage 1, 2, and 3. So stage 4 patients did not undergo operation, so we don't have much of the stage 4 data. But most of the patients are stage 1 and 2 and 3.
And Dr. Na, it looks like we have time for one more question. Let's wrap with this one. What is the response and opinion of clinicians to these technologies?
I think that there are two different response for these technologies. So first response is it is too expensive. So clinicians are very interested in this technology because it can deliver many novel discoveries and novel data for understanding the complex tumor microenvironment. But it is so expensive for a single experiment. So clinicians are not easy to perform these experiment for many samples. So it is kind of very big hurdle for the clinicians. But on the other hand, clinicians are very eager to do this experiment for understanding the complexity of the tumor microenvironment because we currently don't have many knowledge for the cancer progression and cancer evolution based on the tumor microenvironment, the change. So we think that tumors growth and tumors evolution are changing the microenvironment cells and the compositions and their interactions. And so analyzing the tumor microenvironment will give us many information for overcoming the resistance of some therapeutics or to discover new technologies, new therapeutic targets for the cancer. So many clinicians are very interested in analyzing these data.
So, Dr. Na, we have quite a few questions that have come in that are very technical in nature. So we're going to save those to answer via email. So for those of you that have answered those questions, don't fret. They'll be answered to the email address you provided at the time of registration. But I do want to squeeze one more question in here. In the comparison of tumor growth versus fibroblast proportion, what is the tumor size and SUV max features?
The tumor growth and tumor size. So in our study, the fibroblast proportion and the tumor size and SUV max have some correlation. And I cannot get the questions correct. Can I answer this question via emails? Because I think I should rephrase this question for the right answer.
Certainly.
Sorry.
Absolutely. So we'll get to that question via email. And so, Dr. Na, thank you for this excellent Q&A session. Do you have any final comments for our audience?
Well, thank you all for listening to my webinar today. And I am very interested in the spatial transcriptomics. And if you have any questions or if you have some idea for a collaboration, do not hesitate to contact me via my email. And I'm happy to answer for all the questions regarding the spatial omics world. Thank you.
And thank you, Dr. Na, for your time today and your important research. We do want to thank LabRoots and our sponsor, Vision, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today and for their interesting questions. As I mentioned previously, questions that we didn't get to that were maybe more technical in nature. And additionally, those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you for joining us, friends. That's all for now. Hope to see you again soon. Bye-bye.
Thank you.